Hi everyone, my name is Amit Rai, and this is your, I think, seventh lecture. And I've changed the title a little bit. It says Plant Functional Genomics for a Better Tomorrow. Um, I hope okay. Before I begin, this is not the first video lecture you're having. You have heard many professors before through PDF. You have read their lectures through PDFs or video lecture. Uh, Compared to other professors, the difference with me is I'm going to speak only in English. Ideally, when I give my lectures, I prefer to, like all professors, I want to have students so that I can see you, I can judge if you are able to understand or not. So I'm so sorry, this is a very difficult time. We need to keep learning, we need to keep excited, keep interested in science, because only then we can solve world problems like now we have it. So, for example, this coronavirus is one set of problem that is present worldwide, but then it has several associated problems. The the number of people who are sick, the number of people who are hungry, the number of people who have so many other problems, and our as a human being, our approach needs to be uh, to serve as a contributor to solve that problem. So today's lecture is about plant functional genomics, and I hope that the technologies, the knowledge that we have been generating in the past and what we are doing it now together will help us to build a better tomorrow. And it's such a difficult time. It's very important for all of us to remain positive and to think about how we can make our tomorrow better, right? Now, like I was just saying at the beginning of this lecture, that like other professors, I prefer to have my students there, that I can see your face, I can guess if you are able to understand or not. And unlike other professors, I'm mostly speaking in English. So I don't know if I'm speaking too fast or if I'm speaking too slow. So I will try my best to become as understandable to you as possible. But some of you may not understand some of the things. So I have provided my email ID there. Please email to me if you have any question, and we can set up a Zoom meeting or something to solve your problems. Now, in today's lecture, I'm beginning with very few brief concepts and about genomics. I would explain to you what we mean when we say functional genomics, and then I will bring three published papers that I have worked on, and I will try and show to you like briefly how we uh, conduct research to explain, to answer, or to uh, kind of improve our understanding related to processes that undertaken in plant species. And like plants are so important as our food, as medicinal sources, as a factor to control the environment and so on, right? So let's start our lecture. um, Functional genomics I started. So the first thing is, what is omics? It refers to a field of study. Uh, Normally, when we say omics, it refers to a big data set study, for example, which includes a collection of, it's a collective characterization and quantification of pools of biological molecules. So for example, when you say genomics, it's basically study where you are using a pool of a biomolecule, in this case gene, and then you try and understand how things are moving, how their function changes, what's their structure, how their dynamics affects an organism, right? And again, it depends, but it also refers to collective technologies that are used to explore the roles, relationships, and actions of the various types of molecules. So, for example, we say genomics, then this next generation sequencing has played a big role. You cannot imagine, first human genome was proposed in about 1990s. I think Bill Clinton was the president that time in USA. It was like, I think 1990, and it was a big thing. And it took about 10 years till first human genome was published, and it took like billions of US dollar. Many countries took part into it, and today you can sequence one human genome in 3 days. So you can imagine how technologies have moved, how our ability to understand genome has moved, and so on. Similarly, genomics, you have transcriptomics, you have proteomics, and you have metabolomics. And each of these elements provide uh, important information about a specific biomolecules. For example, genomics is about studying gene at a larger scale. And when we say genes, basically what's their function, how they evolved, is there any mutation which changes its function, how methylation of nucleotide changes the regulation of expression, and so on. Transcriptomics is expression of these genes. So again, remember, gene gives transcripts, transcripts gives protein, and these proteins serves as enzymes which results in metabolites. Now, change in levels of metabolites affects a kind of protein response, and these proteins are also transcription factors. They're going to change. Also, they are signaling components. So each one of these elements are interacting with each other. So definitely you can choose one element and talk about it, but they are connected. Therefore, if you try and associate relationships, associate properties of genomics with transcriptomics, and metabolomics or proteomics or several combination, you get a holistic, a complete knowledge of a system. And that helps you to perform or kind of anticipate how a system is going to behave. And that's roughly is what we mean when we say functional genomics. You see, when we say functional genomics, which means we want to understand function of individual molecules, function of individual components. And if we know that, that could help us to decide or design a scheme that could result in what we want to get. So, for example, we want to have more rice. So, what we can do, if we have knowledge about genes which are important to enhance productivity, genes which allow plants to survive, say, salt stress, then we can design our strategy to improve the production. And that's just one example, but these are the similar objectives which drives biological scientists in plant sciences to try and understand function of genes and then use that knowledge to uh, change the system for the benefit of humankind. Yeah, and when I say benefit, just not limited to producing more rice, but it's also, for example, producing more metabolites of medicinal properties, or in improving our ability to grow plants in difficult environmental conditions and so on, right? So a system is a subset of multiple components. So for example, if you see this car has many components. It has a tire, it has a body, it has an engine. Then there are many machine tools which are connected with each other. So when you study tire, you would understand, for example, what type of rubber you need to use, what's the friction, because tire is going to run, how much friction you can control, and so on. That itself is an important knowledge. But then when you look at a car, a better car is when you understand relationship between each of these components, and that helps you to make a car which makes less noise, probably, which are smart, and you can enhance different features of a car by adding or improving relationships, coordination between individual components. But how you can improve coordination between components if you don't know the relationships between them? So functional genomics aims to understand functions and association between individual components. And the objective is to use this knowledge to improve coordination between these biomolecules, to achieve higher productivity or to achieve a desired property in the plant. Okay. And like I said, you have multiple omics technology, and these are connected. And what you can then do is you can perform these omics profiling, perform data mining. These are different tools you can use. You can then perform bioinformatic analysis. You can generate a metabolic model. And what it gives you is a knowledge about the system, which you can then use to achieve a sustainable production of desired trait. And that desired trait could be higher production of rice, that desired trait could be high level of metabolites, and so on. Yeah, so that's the ultimate objective. Now, different people have different strategies. Many people so researchers have different objectives. Now, how to study them? How to study a system's behavior? There are many ways, and of course, I can't tell you everything, it's impossible. But the easiest way that you can actually think of is probably by changing the biological system itself. For example, for example, just take a case. Let's say you are a very, very happy person, and you like listen music, and you are very happy. And then what happens is suddenly this in your house the aircon stops, and the temperature in your house is increasing. Now, when the temperature increases, now your system has changed. Okay. Before, your system was very nice temperature, good music, and very comfortable environment. And suddenly, because of some reason, these comfortable conditions, original conditions, are removed. Now, your system is changed, and this will force you to adapt. This will make you to do some adjustments to face the new challenges. Now, whatever you do, would be called as your response under a given conditions. Now, that response itself tells anyone a lot about what kind of person you are or how you would handle a particular situation. So some people will not care about these changes, but then some people will get probably not so happy, and they may want to go out, and that tells how, and that means different systems behave very differently, right? So in case of plants, for example, let's say this is a picture of Arabidopsis plant. Is a model system in terms of plant functional genomics. This is one of the most important plant. And then the three case studies I'm going to present today is all in this plant species. So how to perform functional genomics in this plant, or how to understand this system. What we can do is we can perturb, we can change this biological system. How? We can do through genetic approach. We can knock out a gene. We can express a foreign gene inside this plant, right? We can also use some biotic or abiotic processes. So for example, we can infect this plant from some insect, or we can add some chemicals. Each one of these factors gonna change the native state of this plant. Now, plant, or not just plant, any system do not like changes, and therefore they will respond. And this responses at system level is recorded through mean of omics analysis. So you can do gene expression analysis, transcriptomics, metabolomics, proteomics. Even in genomics, you can, for example, look at if there is any change in DNA methylation, because change in DNA methylation is an important regulatory event. If a DNA is getting highly methylated, that gene may not be expressed. And these knowledge basically help you to understand the system. What it gives you is a hypothesis that fine, these genes are important. If you do genetic approach, these genes are important. Now that hypothesis is then what you use to predict a system's behavior and then test the hypothesis. This remains a cyclic process. Yeah. So let's begin our first case study. And what's objective? Here the objective is to understand network properties of phenylpropanoid metabolic network. You see, today you're gonna hear about many new compounds, many new metabolites, many new genes. Please don't worry about it. Just focus on how we ask questions and how we answer them. It's not possible for you to remember everything, but you need to know, for example, why functional genomics is important. What's the ultimate aim of functional genomics? Yeah, and how we are asking such questions and trying to answer them. So in this case, in this example, we tried to understand network properties of phenylpropanoid metabolic network. Okay, so phenylpropanoid pathway. Phenylpropanoid is, you can say, it's a metabolite, a family of metabolites which are produced across a diverse plant species. Uh, they have a lot of biological functions we are all familiar with. When you see flowers, they're attractive colors. When you see fruits, when you see different ingredients, when you see flavoring agents, when you see several antioxidants, they are somehow originated from phenylpropanoid. So clearly, Phenylpropanoid has a big role to play for plant to survive different stress conditions, and therefore these are important. Plant also releases phenylpropanoid in the roots. And why that is important? Because when plant roots releases phenylpropanoid, microorganisms take that metabolites as an energy source, and in return they provide several beneficiary compounds. Sometimes they enhance plant growth, and that's a kind of symbiotic relationship between microorganisms and the roots of a plant species. So that's a rhizosphere structure. So phenylpropanoid represents an important class of metabolites. Now, what we want to do is we want to understand how phenylpropanoid pathway is regulated. What's the core regulatory mechanism in phenylpropanoid network, and why that is important? Because if we know, we can perform genetic manipulation to achieve a desired metabolite, right? And the pathway is something like this. Please don't worry about these names, this is just a pathway. Yeah. Now what you see here is these are metabolites. Okay, the arrow represents the relationships. So for example, this metabolite was acted by this enzyme and resulted in this metabolite. Okay, and as you can see here, like any metabolic pathway, these are highly interconnected, they are related, and so on. Now question is how this complex pathway is regulated. What happens if we change this metabolite? What happens when we change this metabolite level? Is plant is going to change its behavior? If it changes, how it changes? So that's the objective. Now, how to study? The easy way is to perturb this system. And for this, we did a very interesting, very smart approach. You see, like I said, plants releases flavonoids, phenylpropanoids in the root. This is different type of compounds released in the root. Flavonoids are, or anthocyanins are kind of flavonoids released by plants in the root. And these metabolites are energy source for these microorganisms. So in one of the old studies, what my lab did was, we, not me, where I did my PhD, student, he studied a compound called quercetin. He identified an enzyme which help these microorganisms to take these metabolites from the root and convert into naringenin. So this enzyme was called QUOA. Now you're gonna listen, you're gonna hear this QUOA many times. Please bear with me. So what that study showed is that microorganisms, not plant, microorganisms have an enzyme called QUOA, and what this enzyme does? This enzyme takes quercetin and converts into naringenin. Now this compound then converts into this, then this, blah, 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 and finally it reaches to the TCA cycle, and that helps microorganisms to get energy. What happens if we remove this enzyme? Bacteria will not grow. Why? Because bacteria needs energy source. From root, it can take energy source only when it converts quercetin to naringenin. Okay. Now so plant makes quercetin and releases in the soil. Now, what we then did, we knew that plants produces quercetin. How? Plants produces quercetin from naringenin. So what we did, we simply take this enzyme and put back to this plant. And we asked a question, what happens? You see, this is the metabolic network, the one which is present in plant. This is quercetin, this is naringenin. So what happens is, naringenin is acted by this enzyme to give this metabolite. This metabolite is then acted by another enzyme which gives dihydroquercetin, and this dihydroquercetin is then acted by FLS enzyme to result in quercetin. Now, once this quercetin is being formed, it is released from its root. Bacteria takes this quercetin here and uses its enzyme QUOA and produces naringenin. Right? So we asked, what happens if we put this enzyme into the plant? So you see, this enzyme converts quercetin to naringenin here, right? And this is a foreign enzyme. Plants doesn't have it. So if this gene goes in, what we would expect, that suddenly this quercetin will start converted into naringenin, isn't it? Because enzyme QUOA takes quercetin as a substrate and produces naringenin. So if you put this enzyme in plant, quercetin, it will take and release it into naringenin, form naringenin. So what means? This means that naringenin level will increase, right? Because more naringenin will be formed. But plant do not like it. It's different from its original state. So plant needs to do something. Either plant can start releasing naringenin, which is not the case, or this naringenin then can move down and then start more and more production of anthocyanins. Remember, more anthocyanin means more colorful compounds. Proanthocyanidins, anthocyanins gives very specific color, right? So means more anthocyanins may form if it goes down. If this goes up, it could affect lignin pathway. Yeah, more lignin means the plant root may have more strength. Not root, plant stem would have more strength. So we did this, we introduced this foreign enzyme. This enzyme belongs to bacteria. We took this enzyme and expressed in plant, and we asked what's happening. Okay, so first thing first, once you express this enzyme, you need to see if there is any difference. Now, expression of QUOA results in increase in naringenin level. What happens if we knock out this gene F3H? If we knock out this gene F3H, naringenin level will, what happens? Because you see, naringenin cannot be formed or cannot be converted into dihydrocamphorol, no F3H, right? Knockout. So if it cannot be catalyzed, it cannot be used by F3H, naringenin level should increase, isn't it? This is very similar to what happens if we express QUOA. The difference is, in this case, this metabolite can go up or down. In this case, when you have knockout, increase in level of naringenin has only one option, go up, but no down. Okay, so we compared this new enzyme expressed in Arabidopsis, we choose to also compare this knockout gene. And then we ask the question, how a system behaves. So once we express this, first thing is we need to check if enzyme is functional. Okay, so what we did, we first created 3 transgenic lines, Q10, Q2, Q11. Q stands for QUOA, so we call these lines as Q lines. Okay, and we first check if these genes are expressed. We saw yes, these genes are expressed in plant. You see the ranking, Q10 is highly expressed, and Q11 was not so high. We also checked enzymatic activity, so we extracted plant protein with QUOA, and we check how much quercetin can be converted into naringenin, right? Quercetin is formed in plant. And again, you can see Q2 was kind of Q2, and Q10 has highest activity, and Q11 has low levels. Okay, so we call them high level plant, low level plant, and medium level, right? 1, 2, 3, rank 1, rank 2, rank 3. So at this point, we confirmed that our plant has this gene, it is expressed, and this enzyme can convert quercetin to naringenin. Then we asked question, what changes we can see in plants? So our research question were, what is network response behavior towards perturbation? Because you remember, we have changed the metabolite level by introducing a foreign enzyme QUOA, or by knocking out a gene called F3H. And both these cases should result in increased level of naringenin. And question is, if naringenin level increases, how a system behaves, and what are the regulatory points? Where, because remember, plants do not like to have any changes. Any system wanna go back to its original state. So what are the regulatory points? And if we know the regulatory points, we can adjust the system the way we wanna adjust. Okay, so first thing, I have already given you this background. So the first thing is that we checked the property. What changes we can see in plants? So our results shows that QUOA activity resulted in anthocyanin accumulation and lignification. You remember that if you may recall the biosynthetic pathway, we have 2 parts. Naringenin level, if it increases, it can go down. Down means anthocyanin. And it can go up. Up means lignin, which is the case when you have QUOA. But what happens in case of this TT6? TT6 is a mutant which stops enzyme from naringenin to dihydrocampherol. So if you knock out that gene, naringenin can only go up but not down. So what we observed in case of plants is, this is wild type. Col0 is the wild type. What we saw is that compared to the original plant, expression of QUOA makes more red color here. Remember, Q10 was the highest expressor. This was number 1. Q2 was number 2, medium expressor. And Q11 was low expressor. What we saw was that the anthocyanin level is increased. Compared to that, TT6 had no anthocyanin, and it's understandable because you see, even if naringenin level increases, it cannot go down. Q10, same case. As you can see here, compared to wild type, Q10, Q2 had high levels of anthocyanin, TT8 has lowest level. So this is about lower path. So what we can say from this data is that expression of QUOA results in increased production of anthocyanin. Probably naringenin goes down and makes more anthocyanin. That's one system behavior. How about upper pathway? So we checked if there is more lignin, and as you can see here, the more color, so we actually use a dye called fluoroglucinol blue. What it does is it binds to lignin. So the more color you see, more lignin you have. So compared to control, you see more lignin in Q10, high expression line. And interestingly, low lignin in TT6. Now, what is TT6? Again, I'm reminding you that TT6 is a mutant line which results when you knock out that gene. And that knockout results in increased level of naringenin. Clearly, naringenin cannot go down to anthocyanin. So where it can go only one place, it can only go to lignin. But to our surprise, lignin was not increased. Rather, it was decreased. So Ref8 is one knockout line which has low level of lignin. And we just confirmed that this is our positive control, and we observed that in case of Q10, high expressor line, transgenic line, the lignin amount is increased. So what's happening in case of QUOA lines is increased naringenin increases anthocyanin, means lower pathway, increases lignin, upper pathway, which is very, very interesting because till this study, nobody could show or could achieve high production of anthocyanin and high production of lignin. You can take either one of them. And this was very interesting. So we asked question, why this is happening? What are the regulatory points? So what you can conclude from this result is that change in or introducing a foreign enzyme changes metabolic pathways. Okay, we also use mass spec analysis, metabolome analysis to check metabolite levels. So this is again, this is a pathway. You remember I keep on using TT6. So this is TT6. When you knock out this enzyme F3H, this enzyme, the knockout line is called TT6. And this means if the gene is not functional, this metabolite would increase. So in case of TT6 line, we thought that increase in naringenin can go up. So maybe lignin pathway will be up, but that was not the case. In case of QUOA, what we observed is that when we have naringenin up, it goes down and makes anthocyanin, which is fine, which is expected. What we also saw was this increase results in upward direction. Lignin pathway increased. So we perform metabolome analysis, and you see this is lignin pathway, this part. This is flavonoid pathway, this part. And these are our Q lines and this TT6 line. This is fold change compared to its wild type, control. Now, as you can see here, the level of quercetin decreases in every case. Why it decreases in case of TT6? If it's a class, I would have asked you. But why it is decreasing is because naringenin cannot form quercetin. Therefore, it's decreased. Why it is decreasing in QUOA line? Because this enzyme takes quercetin to form naringenin. So compared to wild type, this should be decreasing. So in all lines, quercetin is decreased. Okay. Now, how about naringenin? This is your naringenin. What we can see is Q10, Q2 has high level of naringenin. Means this enzyme is functional and this metabolite is small. Remember, Q10 was number 1, best line producing active QUOA. Q2 was rank 2 and Q11 was low expressor line. So level of naringenin was according to activity of QUOA. Is it surprising? No. How about TT6? TT6 knockout also resulted in increased level of naringenin, right? You can see here. But when we looked at other metabolites such as cyanidine, leucopelargonidine, and so on, these two metabolites, you see nothing for TT6. Why? Of course, because this enzyme is not there. If it's not there, then nothing should be produced. Nothing should be produced, right? Now, so this is the overall state. Compared to that, when we looked at lignin, we observed that although in case of TT6, this one, the fourth number, we can see that lignin is little bit the same level or little bit lower compared to wild type. But in the QU lines, all these are increased. Yeah, Q10, Q2, Q10, Q2, Q10, Q2. So we were very interested. We were excited because we can see increased level of lignin, increased level of anthocyanin. Now, you see there is another pathway called phenolamide pathway. So we asked, what's happening to this pathway? So this is phenolamide. To our surprise, phenolamide pathway was downregulated in QOA lines. All these were downregulated. But in case of TT6, it was different trend. In TT6, these were increased. So then question comes, is it this pathway which helps to increase in lignin pathway? Or is it just the naringenin which goes both direction and increases? We don't know. So we next did was we performed expression analysis. This is QRT-PCR, and we found many critical enzymes are downregulated when we compared to the control. And this is for QUOA lines, and this is for knockout lines. Now, of course, it means that expression of a single foreign enzyme brings such a big change in a metabolic pathway. You remember, we just expressed one enzyme, but the whole pathway is changed. Change in metabolites also resulted in change in the expression of these genes, which means plant regulatory system is active and it tries to bring back a system to its control. Then question is how this happens. So what we then did was we used metabolome analysis data, we used transcriptome analysis data, and we map them on pathway. This is called pathway-based integrative omics analysis. Okay. So this is metabolic pathway. Like I said, the names are metabolite, arrows name are genes. These are transcription factors. So for example, if you see this arrow here, means this enzyme is regulated by these transcription factors. Okay, now please notice, black color means no change, purple color means it's up regulated but not too high, about 1.5, less than twofold change. Green means down regulated, red means upregulated. Now, if you just look at this pathway, what you can see is many metabolites are upregulated, many metabolites are downregulated, many genes are downregulated, many genes are upregulated. So what? Isn't it? So what? Genes are upregulated, downregulated, metabolites are upregulated, downregulated. So what? Now, you need to look at very carefully where these changes are happening. All these changes occurring at the junction. What junction? The junction where a metabolite can go to multiple directions. All these changes. For example, in this case, 5-hydroxyferrolic acid can go to form synapate, or it can form 5-hydroxyferroloyl CoA. And therefore, all these junction metabolites and related genes were differentially expressed. And this shows that branch points are crucial for regulation of phenylpropanoid metabolic network. We also observed, very interestingly, that phenolamide may be a bottleneck for lignin accumulation, because in this case what we observed is, in case of QUOA lines, that these metabolites downregulation allowed enhanced production of some of these lignins. So this then becomes very important. Why this is important? You see, there is a benefit, there is an advantage. If a crop can produce more flavonoids and at the same time it can produce more lignin, it helps plants to face stress conditions. At the same time, the value of product increases. So our understanding of network property allow us to predict, not just predict, but design a strategy to improve a crop. In this case, how we learned about a system? We learned about a system by introducing a foreign gene. And then we asked a question as what's happening, how a system is behaving, right? So this is kind of a type of functional genomics which gives you an idea of how a system behaves. And that knowledge you can use for your benefit to design, to create a species which provides you the benefit that you think is important. Okay, now in the second example that I'm going to present, in this case we use the same approach, and this enzyme is also a foreign enzyme, but this is not originated from bacteria. This enzyme belongs to plant. Okay, so what we did, we studied expansion of chemodiversity in Arabidopsis by expression of lysine decarboxylase in Arabidopsis. Meaning, we expressed a new enzyme, lysine decarboxylase. This enzyme is not present in Arabidopsis. This is an enzyme from a different plant. We take this enzyme expressed in Arabidopsis, and then we asked what happens. Can Arabidopsis make new metabolites? What kinds of new metabolites? This enzyme is important to produce medicinal compounds. So, can Arabidopsis make medicinal compounds? Can you imagine a case where, for example, rice, you eat rice, and rice gives you medicine as well. It gives you improved immunity. Again, I'm just giving one interesting, how to say, one interesting imaginary situation, um, that it would be nice, isn't it? Actually, many researchers are working to create plant species which could be used as vaccine, which can be used to produce human protein. I mean, there are many big companies working on it. Okay, so this is a recent study published last year where we try to understand expansion of chemodiversity in Arabidopsis. Now the origin is from quinolisidine alkaloid. What is quinolisidine alkaloid? These are important metabolites produced across plant species. Metabolites such as lupinin, matrine, aspartame, cysteine, these metabolites have medicinal properties like these, and these metabolites are produced across various plant species. Many interesting plant species. Now, these specialized metabolites, these chemical with medicinal properties, kind of provide specificity of chemical properties that these plants have. So, for example, soflavescens has matrine type metabolite dominated, which gives its characteristic, its specific medicinal power. And how these metabolites are formed, this is through a very complex chemical reactions catalyzed by enzymes, started with a amino acid, lysine. Lysine was acted by an enzyme called LODC. LODC stands for lysine ornithine decarboxylase, meaning this enzyme results in decarboxylation of, means removing this CO2, decarboxylation of this metabolite, and it can take lysine or another molecule, ornithine. So it's a dual function enzyme, and this is important. This enzyme can take lysine or ornithine. In general, plants have ornithine decarboxylase called ODC. What ODC does? ODC takes ornithine and performs this decarboxylation to produce putrescine. Putrescine, another compound. I will show you in my next slides. But to produce quinolisidine alkaloid, it is extremely important to produce cadaverine. Therefore, plant species which makes these interesting molecules first need to have this enzyme. And through time, through evolution, enzyme ornithine decarboxylase, through evolution, through random changes, acquired this dual functionality. Once plants got this dual functionality, suddenly this enzyme can take lysine as a substrate and produce cadaverine. Now, what we know is plant which makes these diverse quinoline alkaloids have this enzyme, therefore have cadaverine. But what about plants which doesn't make these alkaloids? Remember, producing cadaverine is not enough. You need to have many other enzymes. So for example, this cadaverine will be acted upon by amine oxidase to give piperidine. This piperidine would then be acted by multiple enzymes to give aspartame, multiple enzymes to give pyridine, matrine, or lupinine type alkaloids. Of course, this means we should not expect by introducing one enzyme suddenly any plant can make it, right? This is normal. We would expect that if we want to make this compound, we need to introduce all these enzymes. So the question we wanted to ask is that the first step is definitely introducing LODC to form cadaverine. But once this cadaverine is formed, how plants derive multiple metabolites? That was the question. So, like I said, the LODC, this is the committed step, means if this enzyme is there, you will form cadaverine. If this is formed, it would result in these alkaloids. Other possibility is this enzyme can take ornithine as a substrate to result in putrescine, which gives a different type of metabolites. So the question is, what would happen if this enzyme is introduced in non-producing plants? Means a plant which doesn't have these alkaloids. Therefore, we would expect different enzymes which are present in producing plants should also be absent, isn't it? For example, in case of Arabidopsis. So the idea was to take this enzyme, and remember, the substrate is amino acid. So Arabidopsis, for example, the model plant already have this lysine. So it's a very simple experiment. What we need to do is take Arabidopsis plant, express LODC into it, check if cadaverine is formed, see if there is any phenotype. You remember in the first example we saw phenotype. So we see phenotype, and then we ask question, what metabolites are formed when you have this enzyme? We also need to check if enzymes or genes are changed in terms of expression level. And then we need to explain how this happened. That's the general workflow for a functional genome analysis. Okay, so it's basically redirecting lysine pool. This is redirecting lysine pool for alkaloid biosynthesis. And here we expected that Arabidopsis has many enzymes. So we thought once cadaverine is formed, several of its endogenous enzymes should make new molecules. We were interested to know if these molecules could be alkaloids. And that was the main objective, that can a single enzyme being introduced could result in new metabolite. Okay, so very similar to what we did, we created a vector, we transformed in Arabidopsis, we collected plants, we then checked if these genes are expressed. So these are independent line names. D-carboxylase is DC, number is independent lines, and we choose 3 lines, DC21, DC29, and DC42. So these showed nice expression of LODC. Again, remember, LODC is a plant origin enzyme, not bacteria. So it's very similar to what I just explained to you, but it's different in the sense that it's a plant origin and we hope to see probably new metabolites. We are not altering a pathway. We just want to see what other enzymes are there which could change this newly formed color. So first thing first, we checked these DC21, DC29, DC42 have expression of LODC, and what we observed is that there is a phenotype. What phenotype? So compared to the control, the root length decreases. Change in root length is often regarded as a kind of a stress. So we observed DC29 had the biggest root length shortening, then DC21, while DC42 was kind of similar, slightly low, but yeah, okay. So we looked at the, if you see at the expression level, clearly wild type has no expression. Why not? Because LODC is not a native enzyme. Arabidopsis doesn't have LODC. Compared to that, DC21 had low level, but expression is there, so we can see LODC is expressed. DC29, the line which showed most effect, most shortening phenotype, had highest level of enzyme gene expression, and DC42, which had least shortening effect, had least expression. So what it suggests? It suggests that the phenotype is correlated following the same pattern of expression here. Same thing was for cadaverine. Remember, LODC converts lysine and amino acid to cadaverine. When we checked the level, we saw that the level of cadaverine was similar to, the pattern was similar to expression level. And lysine is an amino acid, level was kind of not that big difference because it's an amino acid, but we can see that lysine level was slightly down regulated. Star means statistically significant. So at this stage we can say that LODC is working. Cadaverine, a new molecule, is produced in Arabidopsis and the root length has changed. Okay, now remember, LODC has dual function. It can take lysine, it can also take ornithine. LODC can take lysine, it can take ornithine. So we also checked putrescine content and we can see that putrescine level is also increased. Interesting, isn't it? And like lysine, ornithine level was a similar pattern. So at this stage, we can say that we were successful in expressing a plant origin enzyme, LODC, in Arabidopsis. Point number one. Point number two, this enzyme is functional because we can see its expression, we can see phenotype, and most important, we can see new molecule, cadaverine. You see, Arabidopsis do not produce cadaverine. It does produce putrescine a little bit. It doesn't produce cadaverine, not detected. But in transgenic lines, we can see cadaverine being produced. So this new molecule is being formed. Now, question is, after this compound is formed, can this compound be catalyzed by Arabidopsis enzymes? You see, there's a very small difference. The structure of cadaverine and putrescine is very similar, except one CH2 additional in cadaverine compared to putrescine. You see, 1, 2, 3. So 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 1, 2, 3, 4. So one CH2 group is additional in cadaverine. So what we did metabolite profiling. RPLC and HELIC is two different columns. Again, you don't have to go in detail. The point here is when we did metabolite profiling, you see these are DC lines, DC29, DC21, DC42, and this is wild type. What we see is wild type is completely different, and these lines are completely different, meaning definitely in terms of metabolites, these lines has many different metabolites compared to wild type. Another important difference, you see, DC29 is the line which had highest expression of LODC, highest level of cadaverine, and highest level of root shortening phenotype. And what we can see here is clearly this line seems to be very different, very far away than these, suggesting that higher level of cadaverine may result in higher level of many new metabolites. So this results suggested that our transgenic lines may have many new metabolites. How to find them? So one easy way is we performed S-plot analysis. Again, it's not possible to explain everything. If you are interested, please search O2PLS S-plot or S-plot analysis, and you can find many things on Wikipedia. So what S-plot does is it takes two groups. In this case, we use two groups, call and DC lines, and then we can identify features, genes or metabolites which are completely different. Means, for example, the red means these are differential mass features which is in DC lines, and these are specific to wild type. So we used this two different analysis and identified unique features. So, for example, these metabolites were unique to LODC expression lines. As you can see here, this specific metabolite feature is only present in expressing line but not in wild type call. So we took this metabolite and identify through MS based annotation. And we also confirmed if indeed feeding lysine could label them. As you can see here, these are different metabolite names, IDs, their M by Z value, their formula, their annotation, and their structure. As you can see, apart from cadaverine, Arabidopsis also produced 5-aminopentanol, 5-aminopentanoate, valorolactam. This molecule is a very important structure. It kind of gives the ring type structure for alkaloid biosynthesis. It's not alkaloid, and this has never been reported in Arabidopsis. So this is a new metabolite. Similarly, several derivatives of cadaverine. So cadaverine would result, some enzyme should act to give N-acetyl cadaverine. Some enzyme should act on cadaverine to make p-fumaroyl cadaverine, and so on. So we could identify several of these new metabolites. We confirmed them using authentic standards. In metabolome analysis, many metabolites you can predict based on M by Z value, but that is not enough, and you need to have pure standards. So we did that, and we can see and we confirmed them. When we looked at what biosynthetic pathway these metabolites belong, we observed that arginine and proline, phenylpropanoid, and different alkaloid biosynthesis pathway were highly represented in transgenic plants. So our results suggest that a single enzyme in Arabidopsis activated these interesting pathways. Now what it means? It means probably plants in general have enzymes which has multiple functions. It can take more than one enzyme. Now, through some selection process, for example, by introducing LODC, these enzymes starts making new compounds. If these new compounds provide advantage, then these plants would be selected. So that's the evolution mechanism to explain plant chemodiversity. When we looked at the expression of some of these genes, so we identified several metabolites, and in Arabidopsis we already know that these enzymes could convert. So this is putrescine. We know that these enzymes can make these molecules. So we look at expression of these enzymes AT, ACT, AO, ALDH10, A8, A9, and so on. And what we observed is that their expression was slightly increased in our high expressor line. So through this study, what we could show? We could show that a metabolic diversification was achieved. How? By expression of a heterologous enzyme. What enzyme? LALODC. What happened? It took lysine in Arabidopsis and produced cadaverine. So transgenic plants produced cadaverine. But then this didn't stop there, but rather plants enzymes such as AT Arabidopsis and ATA1 helped to produce acetyl cadaverine. We checked, we showed. Cadaverine acted upon by ATCuAO3 to produce 5-aminopentanol. We showed that. And in this study, we confirmed that another enzyme, ALDH10, A8, and A9, helped to convert 5-aminopentanol to 5-aminopentanoid. Of course, we observed many other metabolites, and we don't know what enzymes are involved. But clearly, our results suggested that native enzymes are capable to derive metabolic diversification. And this is a very big step. Why this is a big step is because it suggests the mechanism through which plants derived diverse chemical profile. One single enzyme, which was the committed step for quinolisidine alkaloid, when introduced in plant, or in this case Arabidopsis, native enzyme co-opted these metabolites and produced such a diverse chemodiversity. Such a chemodiversity. And this also means that therefore these new compounds under a given situation may provide evolutionary benefit to the plant, and thus slowly they used this and these enzymes with multiple function to more specialization. And that's how plants started getting specialized in terms of what kind of compounds they are formed. So this study is a way to see how plants evolved biosynthesis of complex phytochemicals. Okay? Case study, we're gonna do integrative omics approach to understand a transcription factor. What this transcription factor does? These transcription factors regulate expression of a gene. And what pathway we are interested? We are interested in phenylprepanoid metabolic network. Okay. Now, in this third and last case study, I have already explained to you phenylprepanoid and a bicentric pathway in the first example. And if you remember, if you may recall this network, you see this arrow refers to an enzyme called the FR. This enzyme expression is known to be regulated by this transcription factor TT8. Meaning, if you knock out this enzyme, this gene will not be expressed. So this enzyme regulates expression of this gene. And then if you have knocked out this gene, no DFR. No DFR means no downward pathway. No downward pathway means no anthocyanin. Therefore, you can see compared to wild type, when you have no TT8, the color of the seed changes. And when you introduce TT8 of a plant, its color go back to wild type. So this transcription factor was very well studied. It was very well known. People know what this transcription factor does. So question is, what's new? Why we still study on this gene when everything is known? No, everything was not known. And that's the beginning of this story. So you can see here, this is a knockout line and this is a control line. What's very interesting to us was, other than blocking the whole biosynthesis pathway, we observed that, for example, flavonoids are known to have many sugar molecules attached. So different types like Q1, Q2, Q4, Q8, and so on. But when we knock out this gene, we saw no sugar is attached with this. Q stands for quercetin, it's a metabolite, Q, and peak 1 is different glucosides. Okay, what we observed is one transcription factor completely changes these complex metabolites to a single metabolite, which is Q. Now, what we knew was that it regulates downstream, means BAN and DFR, means no anthocyanin. But what we were surprised to know is, wow, many sugar conjugates are gone. Question is why? We thought maybe TT8 is doing something else. So we perform metabolome analysis. This is exo, this is endo. And we can see, for example, many peaks are disappeared compared to control. You can see many red ups. So what we said, fine, metabolome analysis shows that removing TT8 results in downregulation of sugar conjugates, glycoconjugates. Why? So to know that, we performed a microarray analysis. Microarray is a transcriptome approach. Integrative omics workflow was something like that, where we used metabolomics, transcriptomics, and genomics. Different analysis gave us different results. So first we analyzed independently and together. And then we combine these data sets to get a biological interpretation. So first we performed expression analysis. This is control, this is knockout. And as you can see here, we observed many genes are up in TT8 and many genes are down in TT8. We could identify enzymes which are involved in attaching sugar molecules to metabolites. So very good. We were very happy and we reported that these enzymes are important. Now, to us, what was even more interesting is when we looked at these genes, the enzymes which are highly upregulated in TT8, we observed all of them are associated with stress conditions: abiotic stress, biotic stress, phytohormone, metabolic pathways, developmental processes, which was very interesting. You see, abiotic stress is highly upregulated in TT8 compared to downregulation. Phytohormone was, wow, you can see here, jasmonic acid, salicylic acid, abscisic acid, all was highly upregulated. Now, this is an important indication. Phytohormones are important component which activates biosynthesis of secondary metabolites. So, meaning somehow directly or indirectly, this TT8 is associated, related with phytohormones. So what we did, we choose transcriptome dataset, we used metabolome dataset, and we did integration. So what we realized is that 1, 2, 3, 4. These 4 pathways are significant in terms of metabolites and gene expression is affected. So green means downregulated, red means upregulated. So in this case, for example, this is jasmonic acid, one of the phytohormone. You can see the enzymes are upregulated and metabolites are also upregulated. Clearly, it means that the gene's expression and the effector molecule, meaning the genes gives protein, protein gives metabolite changes, right? So both genes and metabolites are changed. This was very, very interesting. So because we know that this is a knockout line, TT8 knockout line. Now, if TT8 is not there, all these enzymes showed high expression. Means maybe TT8 is a negative regulator. Now, TT8 somehow interacts with these enzymes. So, therefore, we thought, you see how a transcription factor regulates the expression. Transcription factor goes to the promoter, binds to the promoter, and then regulates its expression. So clearly, our hypothesis was that genes which are affected in the knockout line should have some pattern in terms of promoter similarity. So we performed a promoter similarity analysis. So you can see here, this network is based on promoter similarity, and the different shapes represents different types of processes. We were interested in what processes are kind of linked with TT8, and this analysis allowed us to identify many candidates which is directly linked with TT8. That helped us, this promoter analysis helped us to identify possible relationships. So you remember how we did? We first performed metabolome analysis that suggested that knocking out a transcription factor affects a metabolome. How? So many metabolites are downregulated and sugar molecules are not conjugated. So what then we did? We performed expression analysis, transcriptome analysis, and transcriptome analysis showed, yeah, many genes are affected. Significantly, genes related to phytohormone biosynthesis is also affected. We combine these two datasets using integrative omics approach. What type of integrative omics approach? In this case, we are choosing individual analysis and then mapping to the pathway. Yeah, so it's basically the approach. In the previous slides, I explained to you. When we put them together, we realized that these pathways are high. Then we use differential genes, genes which are expressed in different levels, and looked at if their promoters are similar. And we found, yes, they are similar. So then we believed, our hypothesis becomes that TT8 interacts with these enzymes. So we selected and performed first gene expression analysis. This is a bit complicated. I would recommend you to read the paper. This is published data. But what we did was we created a inducible system where, so for example, you see all these genes you see is reverse. So this black is a small, then this gray is high. And if this black is high, this gray is small. Why? So this is comparison of knockout line versus wild type. And this is a overexpression line, means in this case TT8 is highly expressed. So this analysis helped us to identify genes which are low expressed in TT8 when it's gone and high expressed in TT8 when it's very high. So this helped us to identify many genes, and then we perform CHIP analysis. CHIP is a chromatin immunoprecipitation analysis. Please search Wikipedia. You would learn many things from there. The point is, based on expression analysis and CHIP analysis, we identified several genes. So BAN we already knew, but 1, 2, 3, 4, 5, 6, 7, 8, 9. We identified these 9 new relationships with TT8. If this is a high, meaning this region TT8 binds. So it's a physical interaction, kind of an interactome, protein DNA interaction. And this suggests that TT8 physically binds here. Okay, so what we then did was, because all those genes are related to stress, we asked if TT8 knockout is not good under stress conditions. So we used a pathogen. It's a plant pathogen called P. serine J. Is a kind of bacteria. What we did was, this is TT8 and this is wild type. Uninfected are growing healthy, but when we infect them, we observe that knockout line dies immediately, very quickly. Compared to that, wild type still survived relatively better. So then what we did, like I just explained to you, we created an inducible overexpression line. When we induced the expression of TT8, we observed that, wow, this overexpression improves the chance. You see, this is healthier plants, which means that when TT8 expression is high, plant is able to handle stress better. If you are interested to understand how we created this inducible line, what is DEX and MOC and so on, please read this paper. It's already published, and I think it's a very good example where you can learn how integrative omics, how multi-omics analysis assists to understand new biological insights. And this is the final model that we proposed. What we observed is that one transcription factor, TTA, regulates not only sugar conjugation machinery, but it also regulates stress response processes. And therefore, this sugar conjugation machinery is as important part of plant immunity as these genes, as these phytohormones. And therefore, our results suggested that this transcription factor is important for plant species to regulate how to respond under a given condition. Now, this understanding is important. Why? Because then it allows us to prepare our strategy when a plant is under for example, we know that global warming is increasing temperature worldwide, and the increased temperature is also a stress to plants. So, such functional understanding allows us to prepare our strategy to improve plants' crops' tolerance under stress conditions. Okay, so with this we come to our last slide. If you have any question, you can always email to me, and I will try and organize a Zoom meeting. We can discuss on that. I think this lecture has about 90 minutes, which is normally what your normal lectures are. So if you have further questions, please write to me. With this lecture, I'm also going to upload an assignment question. This is very easy. You simply have to answer what is functional genomics, or why functional genomics in plant sciences is important for better tomorrow. That's the topic. That's the lecture name. So that's the only question. Try and answer in English, of course. Whatever you could understand, if you think it's important, why it's important, try and explain in your answer as much as you can. Yeah, so that's about it. I hope through these three examples you could see how we can change a system, and once we change the system, how that helps us to understand and perform this functional genomics. And these knowledges are very important to improve the crops, to improve our understanding of a plant system, and so on. Okay, so with this, I'm ending this lecture. Please let me know through your comments if there is something that you want me to explain further. I have tried my best to speak slowly, but of course, few places I would have been a little bit faster or not so good to understand, so my apologies. And yeah, if you listen this lecture a few times, probably you will understand better. Okay, so thank you so much. I wish you all a very healthy learning, and hope soon this whole thing ends and we can resume our learning the way we used to have before. Okay then, take care.